Hello all. Something's gone seriously wrong if Ebola becomes part of your routine practice. But I find it very interesting, thankfully, because it's been most of my life for the past 6 months. And I think it is interesting. It teaches us a lot about vaccine research in general and what can be done on the national and international stage in terms of responding to epidemics and how we might respond to this epidemic and future epidemics. So just where it all began was back in 1976 when there were two outbreaks of a previously unseen disease that was highly fatal. One in Zaire, well, former Zaire, now it's Congo, and the other one simultaneously in Sudan, with the Zaire outbreak near the Ebola River, which is where the virus gets its name from, having a 90% mortality, which was and causing a spectrum of viral hemorrhagic fever, which I'll explain a little bit more about later on. So the photo on the left is one of the wards at the Yambuku Mission Hospital, I believe. And on the right, there's a chap there in a very snazzy shirt who is Peter Piot, who was then a 27-year-old Belgian research scientist and now is the head of the London School of Tropical Medicine, who was part of the team that initially discovered the Ebola virus. So as the story goes, they were sent a thermos flask with vials of blood from Kinshasa in Zaire with a sample of blood to be tested for yellow fever, with the story of what had happened. It was from a nun who died of this strange disease. Was it yellow fever? And I doubt it was as pretty a picture as this that he saw, but essentially this is what he was confronted with down the microscope, which was this massive virus. So I've put a measles virus there for comparison. So it's a filamentous, wobbly, 980 nanometers long virus, and much bigger than what you'd previously see. The only thing that it even remotely resembled was something called Marburg virus, which had been discovered about a decade previously in an outbreak associated with research scientists in Germany and Yugoslavia, which had caused a hemorrhagic fever. So initially it was thought maybe this is Marburg. It's moved on slightly from there, where we've actually realised that there's a family of filoviruses, three parts of which, so you've got the Marburg virus, Ebola virus, and Cueva virus, which is not known to be causing any disease in humans. It's been seen in bats in Spain, I believe. And the genus of Ebola split into 5 species. So there's the Zaire Ebola virus, named after the original outbreak in Zaire. Sudan Ebola virus, which was the simultaneous outbreak in Sudan. And then 3 other Ebola viruses, which have of descending importance to humans as they go down in terms of their lethality rates. So Zaire, at worst, Zaire Ebola virus having a fatality rate of 90%, although we're seeing slightly lower mortality rates in the current outbreak. Sudan, around 50%. The Bundibugyo virus has been seen in Uganda, I believe the case fatality rates there are about 30%. As far as I'm aware, there's only been 1 case of Ivory Coast Ebola virus or typhus virus, and that was they survived. So that's got a fairly low mortality rate. And Reston virus, which is only associated with animals, so that's been seen in research, non-human primates, etc., but not known to cause human problems. This is the structure of a filovirus. Not to be too confused, the only thing that really is of importance to most people is the glycoprotein there. So this is essentially the only significant surface protein that you've got on the Ebola virion, which has been why it's been the target of many vaccines and also any treatments. There's only 7 proteins in total on the whole Ebola virus structure, and it's a single-stranded RNA genome, which is listed at the bottom there, with about 19,000 nucleotides. So this is just illustrating the clinical syndrome that's typically associated with Ebola. Sorry if it's not projected all that well, very small writing, I'm afraid. So essentially, it has a variable incubation period, Ebola, of anywhere from 2 to 21 days, but the median time being about a week. Your initial symptoms would usually start about a week to 7 to 9 days in. It would be fairly non-specific and flu-like, headache, fatigue, fever, muscle soreness, but it progresses very rapidly over the subsequent week with hemorrhagic complications, impaired consciousness, and as we've frequently seen, death. And there's no licensed treatment available beyond supportive care, which is, I believe, what Rachael will be talking about later on. The life cycle of Ebola represents what we currently understand. So it's almost universally agreed that the reservoir of infection is the fruit bat in Africa. That's because it's, you can see asymptomatic Ebola infection in fruit bats, and early sort of epidemiological studies would point to the fruit bat being the carrier. Then you have circulation amongst other animals, including non-human primates, who it also will cause a highly lethal outbreak in. And then from the wildlife epidemics, you then get infection of humans, and within humans there can be human-to-human transmission, likely affecting healthcare workers, burial workers, and those that are caring for the sick and unwell. So I think this is quite a powerful infographic in terms of expressing the scale of the current outbreak. This is from The Economist. So the bottom line shows these tiny little blips are all the previous outbreaks and the years that they happened. So you can see why not much attention was paid to Ebola. And this whopping great line here is the outbreak that started from December 2013, with the darker section being deaths, about 10,000 deaths, and the lighter section illustrating the caseload of about 24,000 cases. And this is really representing the scale in the major affected countries of Guinea, Sierra Leone and Liberia, again with the darker areas representing deaths, the lighter areas being caseloads. These are obviously estimations and they'll be imperfect, but I think it really does sort of somewhat get the point across. So it's estimated that at least 60 times, so this outbreak is at least 60 times the caseload of any previous outbreak. So in the face of this, there's been a massive international effort to expedite vaccine and treatment development, and also to get field workers onto the ground. So I'm going to just focus on vaccines from here on in, because that's my area of expertise. So this is the typical timeline of a vaccine development, all the way from identifying a disease, identifying antigens that you might want to form a vaccine against. So this is all very basic research science, up into preclinical testing, animal testing, test tubes, etc. And then this is when you start with human vaccines, the phase 1 studies, all the way through into a licensed vaccine. And you can see from the timelines at the bottom, this whole process can take anywhere from 10 to 15 years, sometimes longer in the case where you're having to make a case for cost benefit, etc. Fortunately, at the start of this outbreak, much of this early work for us had been done. So there were a lot of preclinical work had been done, and there were vaccines that had been tested in animals, etc. But we still had this huge chunk to go before we might get a vaccine on the ground. So you can see we're trying to condense sort of 5 to 10 years minimum work into a very short space of time. The aim was to get these vaccines on the ground in Africa within 6 months to a year. So it's required a massive investment of time, massive commitment from pharmaceutical companies, academic institutions, regulators such as MHRA, and governments. So the current vaccines in development, so I'll go through them in order, although I'm working on one. You might be able to see my bias in the way I express it. Hopefully not. I've tried to be as unbiased as possible. So the first vaccine is the GlaxoSmithKline vaccine, which was heavily publicised in September of last year, which was started a phase 1 trial here at this site with the Jenner Institute in September 2014. So that is using a chimpanzee adenovirus, much in the same way as Chris was referring to with the RSV vaccines. So it's a replication defective chimpanzee adenovirus expressing the protein from the glycoprotein from the Zaire Ebola virus. I will just go on to explain exactly what a vector vaccine is. So this is the life cycle of an adenovirus, grossly simplified. I've made it myself to explain. That's the little adenovirus there. It looks like a moon lander there. So it wants to find a cell, be absorbed, enter that cell, hijack the machinery of the cell to make lots of copies of itself, and then express some of its own internal proteins within the surface of the cell. Then make copies of itself, lots of little moon landers there, and then release those copies and go off to infect lots of neighbouring cells and start the cycle over again. So with a vector vaccine, what we essentially do is we make one genetic modification, which is to stop that viral replication. So you stop it only at that stage there, so you actually still hopefully get some protein expression on the surface of that initial cell that is entered by the vaccine. And then the second genetic modification you make is that you get it to express your target protein. And then you're conning your immune system into developing an immune response to that particular target. Fine. So this is showing results from that initial Jenner Institute trial. I won't labour on it other than to say that it looks like there is immune responses here. So this is showing that there's antibody responses after vaccine, and this is showing that there's T-cell responses after vaccine, which is promising. It was also reported in the New England Journal as being safe and tolerable to the adult volunteers. They started with just that single chimpanzee adenovirus, and then, as Chris also alluded to, it's very frequently with an adenovirus vaccine you will also give a modified Vaccinia Ankara vaccine. So they've boosted recently with this particular MVA vaccine, which is a modified smallpox vaccine. And this doesn't just express proteins from the Zaire Ebola virus, but also from the Sudan Ebola virus from Marburg and Thai forest virus. So it's hoped that it wouldn't just protect against this outbreak, but future outbreaks as well, if it's effective. So the next vaccine to discuss is the Merck vaccine. So that was initially developed by the Public Health Agency in Canada with technology licensed to a small biotech company in the States called Newlink, who were then had an exclusive licensing deal with Merck to develop the vaccine. And this is using a recombinant vesicular stomatitis vaccine, which is live and replication competent. So some advantages of using a replication competent vaccine is you hope it will create a more significant immune response, but concerns about it would be that it's potentially more reactogenic, you might get more fever, you might get more significant symptoms. Clearly having a fever in Sierra Leone or Liberia could be problematic in that you might very well be assumed to have Ebola and taken to an Ebola treatment centre, and if you didn't have Ebola by the time you arrived, you probably would by the time you left. So that all needs thinking about carefully. And the third one, I'll go on, I'll tell you, this is my vaccine trial, which is why there's potentially more detail there. I'm not trying to bias it, I just know more about it, unfortunately. So this is very similar to the Jenner trial, in terms of using an adenovirus and an MVA vaccine. We're using a human adenovirus vaccine, adenovirus 26, which is a Johnson & Johnson vaccine. The study has been sponsored by Crucell. So we began that phase 1 trial in December 2014. We very quickly enrolled all 87 participants, and we've given all the first doses of the vaccine. And to explain, with this particular study, from the start, we were always intending on using both vaccines in combination. So everybody's going to receive 2 different vaccines, apart from the placebo group, obviously. And that's because the hope is that by using a combination of vaccines, you'll produce a more durable immune response. No data yet to show you, but watch this space. We'll have it out by May. We're hoping we'll have some data to report, but no major safety issues to report to now. And then we need to take these. How am I doing for time, by the way?
I think you probably are nearly
Okay. Fine. I will just rush the last bit then. So we need to take these vaccines to Africa to develop them and to prove they actually work, because even if we can show that you produce antibodies with the vaccines, we don't actually know in the real world if they're going to work. So how are we going to do this? So the current plans are Prevail, the Partnership for Research on Ebola Vaccines in Liberia, Strive, the Sierra Leone trial to introduce a vaccine against Ebola, and then whoever was writing acronyms ran out of energy. Or maybe they're still writing the acronyms. Maybe they'll be really good. And I'll just explain very briefly the strategies there. So the Prevail trial is in Liberia. They're aiming to do a randomised placebo-controlled trial and enrol a massive 27,000 adults, including risk groups, but aimed at the general public. And they're going to give 9,000 people the GSK vaccine, 9,000 people the Merck vaccine, and 9,000 people placebo, and then observe them for 8 to 12 months and see if they get Ebola. So that will be interesting. The ethics of giving someone placebo in this situation has been hotly debated, and there's differing opinions in different places as to how to proceed with that. But certainly our tradition would be that the best way to show that it works is to use placebo. But whether or not you will have people willingly subject themselves to possible placebo is another matter. Excuse me. The Strive trial is using the Merck vaccine, so that's essentially targeting healthcare workers for vaccination. And the way that they are going to set up a control arm is that they're going to randomise people to either get the vaccine there and then and observe them for up to 6 months, or they get the vaccine at the end of the 6 months, which is how they're getting around the issue of it's not ethical to not give someone a potentially very helpful treatment. But they're still delaying that potentially very helpful treatment, but that's the way they've gone. And the deferred vaccination group can act as the comparator arm for the duration of the study. And then the last strategy is the concept of using ring vaccination. So you identify cases of Ebola and then you vaccinate around them, so they're close contacts, in the hope that you prevent onward spread. And that's similar to what was used in the smallpox eradication scheme. But again, you have someone who's had contact with Ebola, has a fever, they'll go to the Ebola treatment centre. So it does bring up some difficulties, and I think I'm being waved at frantically, so I think I've said enough, and I'll pass you on to Rachael to discuss her aspect.
So my name is Rachael Cummings, and I'm the Senior Humanitarian Health Advisor for Save the Children, and Sarah asked me to come and talk to you today about Save the Children's response to Ebola in Sierra Leone. A bit of background, I'm a nurse. I trained way back in Newcastle quite a few years ago, and have worked various places, but have worked for Save the Children now for about 6 or 7 years as a humanitarian health advisor. I went to school with Sarah, so that's the link with Sarah, just so you know how I arrived here. So obviously back in May, the first few cases in Sierra Leone out in the east, in Kailahan, appeared. Save the Children's been working in Sierra Leone since 1999, so we had an established office and we'd already started supporting the Ministry of Health and the government to respond to the outbreak. However, we were aware as the organisation that we needed to do more, so we were supporting contact tracing, we were supporting healthcare workers, we were supporting communities with social mobilisation, but as an organisation with humanitarian health ambitions, we were very aware that we needed to do more. MSF were crying out for help. MSF were at the brink of their humanitarian capability in their response. So we had a discussion with the Sierra Leone government, who'd already allocated land in Kerry town, just outside of Freetown, for an Ebola treatment centre. Kerry town became a very well-known and famous treatment centre for many reasons. One of them being that the president of Sierra Leone sort of placed his stamp on this and said, okay, Sierra Leone needs a big treatment centre, we're going to put it in Kerry town. Save the Children were requested to do it, and we did a lot of soul searching as an organisation, because up until 6 months ago, we had never done Ebola. We'd never worked in Ebola, we didn't understand Ebola. We hadn't really done hospital care, we hadn't really done secondary level or tertiary level care, so it was a massive step change for our organisation. However, the humanitarian imperative to do more as an organisation with humanitarian health ambitions was really driving us, and of course the needs on the ground were huge. I went out in September as the feasibility team to look at how feasible it was for Save the Children to actually run an Ebola treatment centre. And already September was too late. We were behind the curve in terms of the outbreak, but the outbreak was, increasing by the day. So the decision was made to open and support the Ministry of Health and the government of Sierra Leone to run Kerry Town Ebola Treatment Centre. Now, the objectives of Kerry Town, or the objectives of responding in an Ebola outbreak, are pretty basic. You need to remove people from communities to break the transmission. That's the key, and it's a nasty disease. It has an impact on families, has an impact on communities, and it's nasty in that you have to remove sick people from the people who want to care for them. But in order to break the transmission, you need to do that. But what we also decided as an organisation with humanitarian health ambitions, having never done Ebola before, is that we were going to use not only a public health approach, which is to move people from communities, but also providing a very high level of care for patients in the Ebola treatment centre. So I'll show you later some of the outcomes of our interventions in terms of our case fertility rates for the patients that arrived. And also, restoring confidence in the health system. We had, as an add-on to our facility, a 12-bed facility which became a 20-bed facility, which was specifically for healthcare workers from Sierra Leone and internationals, and that was run by the Ministry of Defence. So the UK military were on site in Kerry Town working with Save the Children to provide this outbreak response. Now, Save the Children never worked in Ebola, never worked really in a hospital. We needed to develop very quickly, as the infrastructure was going up, a capability of our own to be able to care for patients safely, and also to make sure that our staff were going to be safe. So we had to develop our clinical guidelines, our standard operating procedures, which includes taking off your PPE, putting on your PPE, all of your equipment, all of your supplies. And we did that by working with other agencies. So MSF obviously have got the groundwork done. WHO have also got their standard operating procedures. But again, Save the Children wanted to improve the outcomes for patients and ensure the safety of our staff. So we took it very, very seriously. And again, having never done this before, we were very aware of the risks as an organisation, and also the risks on the individual who was going to come and work with Save the Children. Now, we had many partners working with us. I already mentioned the Ministry of Defence, the UK military were there with us, or still are there with us, it's still open. The government of Sierra Leone and the Ministry of Health were the first main partner. We had the Cuban Medical Brigade as a foreign medical team working with us via WHO. We had Public Health England, who were running the laboratory on Kerry Town, still are running the laboratory in Kerry Town, who've been amazing, I have to say. And then we had Save the Children and the membership of Save the Children also providing us with clinical staff, logistics staff. So there's many, many different partners having to work together. And our role as Save the Children was to ensure that everyone was following the same standard operating procedures to ensure the safety of all staff, which would eventually improve the outcomes for our patients. So it's quite a complex beast. And this is just a picture of some of the training. We've got over 600 staff working. So you imagine it's a hospital that happens to be treating one disease. And that disease is very, very deadly. It's not particularly contagious, but it's very, very deadly compared to other diseases. So the case fertility rate can be very, very high. Healthcare workers have taken a massive hit in the outbreak, especially in Sierra Leone. Over 500 healthcare workers have been infected, and that's basically because they weren't equipped to be able to manage the diseases that they were faced with. And especially early on, people just didn't have gloves, they didn't have the knowledge, they didn't have the chlorine to be able to protect themselves. In the training, the first thing we teach people is in Ebola outbreak, you have to protect yourself number one, you protect yourself. Number two, you protect your team. And only then can you treat the patient. And as a nurse, as a healthcare worker, that goes against the grain, really. Your initial response is to help people, but actually your initial response in Ebola has to be to help and protect yourself. So it takes a lot of understanding to be able to ensure the safety of the staff, which in turn ensures the safety of the patients. This is a picture, just a photograph of the laundry area at the back of the treatment centre, and it was just to give you an idea of the scale of the operation, which again, it's something Save the Children hasn't done before. So it was very much learning on the job. But the suits that we wear, we were getting through about 1,000 suits a week, and they are destroyed. So you have to think about the whole supply chain of equipment, drugs, medicines, equipment, etc., that you have to be able to get at the right place at the right time so that the nurses and the doctors and the clinical staff can have all that they need to be able to provide that care. Now, the NHS staff that joined us were, I'm just aware of the time, the NHS staff that joined us in December. We opened a very rocky opening at the end of October, November, which the BBC filmed for us. And the NHS staff joined us in December. And I have to say the impact that they had within 24, 48 hours, we could have a visible and a tangible difference in the level of patient care that was being provided. It was very, very interesting, just the basic nursing care that's needed to ensure better outcomes for patients was tangible within 48 hours of NHS nurses, actually, rather than the doctors. It was the nurses who were willing to really get in there and provide basic nursing care. So this is a picture of two guys going down to the wards, and they're carrying drugs and they're carrying water bottles. And we had to start to think, if you've got 50, it was a 100-bed unit, if you've got 100 patients, all of whom we have to get to drink 6 litres of water a day. Not all of them can drink, so say 50 people are drinking 6 litres of water a day, or ORS. How do you physically get the water? There's no piped water, there's no tapped water. So we literally had to have human conveyor belts of people carrying water, and again with food. And it was just this constant rethinking of how you would normally operate in a hospital, but you had to start to second guess everything that was required. Okay, I've got some graphs. I wasn't sure who wanted to see graphs, but this is, I quite like graphs. But so we opened on the 25th of October, 27th of October. We then closed because it was very, very difficult. This is where we were learning the majority of our lessons in how to manage an Ebola treatment centre. And you'll see that the majority of these people are confirmed, so we were only taking confirmed cases in the beginning. So we knew who was going to be coming through the door, we knew how to manage them. And mid-January, we opened the doors to anyone, so suspects, walk-ins from the community. But you'll see the number of confirmed dropped dramatically, but the number of suspects, so people just coming in from the street, increased as well. And the walk-ins, people just literally accessing it from their houses, continues and still continues to this day to increase. The outbreak is by no way over. And for the suspect patients, so those people that were coming either via an ambulance because they've got a fever, they've got a headache, they were feeling general malaise, or they were coming straight from their homes. You see, the majority haven't got Ebola, but the challenge then becomes when you've got a crumbling health system, where do you send people to? Because you don't want to keep them, because you don't want them to catch Ebola, but where do you send them to when the health system is pretty much on its knees? So there's constant challenges that we're still facing. So I said at the beginning, one of our focuses was on patient outcomes. So MSF took very much a public health approach, we took a public health approach and a patient outcome approach, individual patient outcomes. And you'll see how we have improved in terms of our outcomes for patients, but also this tells only part of the picture because it doesn't tell you who's coming through the door in terms of what stage they are in the disease. If people are arriving early, they've just got their temperature, they're just showing some signs, then chances are they'll have a much better outcome than those patients who delay seeking treatment for various reasons, and then they have a much poorer outcome. And in March, we've only had very few cases who actually came very late on in their disease progression. And just to say, the challenge is continuing. You know, this week there's been over 30 cases already of Ebola in Sierra Leone. We're a lot behind Liberia, who's now had 19 days, I think it is, without a confirmed case, which is fantastic. Sierra Leone has constantly followed the trajectory of the disease in Liberia, but still we're yet to see the decrease and the sharp drop that Liberia has seen in the outbreak. We're very much working, you know, to get to zero, but to remain at zero in terms of the outbreak. And so the children's role, once the Ebola has finished, is very much to stay around and support in terms of the health system strengthening and the building of the primary healthcare units, which is where we were working prior to the outbreak. And I think that's it. Thank you.